On the other hand, for the case of response elements for thyroid hormone, retinoic acid, and vitamin D, we will be able to notice that the DNA sequence is actually the same for the three hormones, and the difference lies on the spacing between the half binding sites. Right, so letter N on the middle represents the spacing. So the space is particular and determinative of what hormone will be binding to such response element. If the spacing is 5, we can say that it's a response element for retinoic acid. If the spacing is 3, then the response element is actually specific for vitamin D. No? Now, so much about how group 1 lipophilic hormones function in the process of response creation. Now, let's consider the same case but for group 2 hydrophilic hormones. A while ago, we have described hydrophilic hormones as water-soluble hormones that have no transport proteins. Hence, their plasma half-lives are very short relative to group 1. More so, the case necessitates that the receptor for hydrophilic hormones be found on the surface of the cell because these hormones cannot traverse the length of the plasma membrane. More so, we've described a while ago also the different secondary messengers or intermediate molecules that ensure the propagation or non-termination of signal until a cell actually responds in the presence of the hormone. No? A few examples of these secondary messengers include cyclic adenosine monophosphate, cyclic guanosine monophosphate, ionic calcium, phosphatidylinositides, diacylglycerols, no? And as we've described a while ago, there are group of hormones that can use these secondary messengers. Now, to understand how hydrophilic hormones participate in the process of response creation, the process of producing signal transduction, let's take into account the the case of G proteins. Now, but before we talk about G proteins, let me introduce to you receptors for a very popular group of receptors for hydrophilic hormones. Let me introduce 7-TMS receptors. 7-TMS receptors stands for 7 transmembrane surface receptors. Right? 7 because the receptor actually contains 7 alpha helical domains. So you can count the cylinders 1, 2, 3, 4, 5, 6, 7. So these 7 cylinders represent the canonical alpha helical domains, no, of the 7-TMS receptor that is important for recognizing group 2 hormones, for recognizing hydrophilic hormones. It's important to take note that it's not the receptor alone that is important for the process of signal transduction to occur as the hormone binds to it. You are able to observe also two other structures that we are yet to identify. These two structures important for producing secondary messenger by the hydrophilic hormone. So these two structures are our G protein, our multi-subunit G protein, okay, which is bound to guanosine diphosphate (GDP), and an effector molecule that can be in the form of enzymes, no? Like for the case of the enzyme producing cyclic adenine or cyclic adenosine monophosphate, no? Let's talk about how the 7-TMS induce alongside with the G protein and the effector molecule induces signal transduction once a hydrophilic hormone binds to it. Right. On the left, you are seeing an inactive status or inactive representation of the components. Alright. On the right, you're seeing the